right, we're at the top of the hour. I think we'll get started. Hello and welcome, everybody. My name is Martin Myers, and I am on the board of the Society for Immune Polymorphism, and I'm excited about this webinar. I wanted to just tell you a couple things about the Society for Immune Polymorphism. This is our website, immunepolymorphismsociety.org. And our mission is we're an international membership organization of scientists dedicated to understanding the genetic and functional variation of the vertebrate immune system and the immune system's role in evolutionary biology, disease, and health. Although the talk you're about to see is making us reconsider our mission because I think we need to look beyond vertebrates and kind of expand. There's some interesting things on this website. Our last three annual reports, and we've had multiple webinars. They're all archived if you're interested. We have annual symposia, which you will hear more about if you get connected to us. And oh, I forgot to mention our board. We have three new board members, so now we have 12, and want to welcome Tiziana, Katie, and Tobias as our three new board members. And last but not least, please join the society. You click here, immunepolymorphismsociety.org, join us. And there are some benefits of membership listed here, and there's more to come. So I would like to invite Yuko Ota to moderate the session because she knows a lot more about this area than I do, and I think she's known Tony for many years. I'm fascinated by it. I probably stand to learn the most here. But Yuko's been working in this space for a while, comparative immunogenetics, MHC, and is working with some interesting model organisms like shark, which we were just talking about. So I will have her introduce Tony, and then when it comes time for Q&A, she will hopefully be able to help navigate that. Please put questions in the chat at any time. Thank you. Yuko, take it away.
Thank you, Martin. And good morning, afternoon, and evening, wherever you are. And this is my great pleasure to introduce Tony Di Tomaso. I have known him over 30 years now, right? 30 years. But all this time I've known him, he was working on this C-squat model, the colonial tunicate. But I was reading his bio, and then he went to Stanford for undergraduate, and he was exposed to the marine biology and then excited. But then he went to graduate school for mouse work, Washington University at St. Louis. And I was pretty surprised because I never heard him talking about the mouse. And after he graduated, he came back to Stanford and worked under Herb Weissman. And then he had the tunicate colonies set up at Hopkins Marine Station. So he took that project, very brave, I think, very brave. And then he became PI there. And 2009, he moved to University of UC Santa Barbara, and he started from assistant professor to now he is a professor. So he works on this colonial tunicate and then works on all different aspects of this animal model to a mammalian system. So for example, he has stem cell biology, aging, and then as today's topic is non-self recognition. So non-self recognition is, of course, the immune recognition, first step of immune recognition. So I'm also working on the evolutionary aspect of origin of adaptive immune system using shark, and mainly shark MHC and then other immunoglobulin and T cell receptors. And we work on evolutionary aspect of immune system, but we have to understand the mammalian system. So we are not working on a different aspect of immune system, but we are getting additional aspect of immune system into this mammalian system. So combining all this mammalian system and then other lower vertebrate, or in this case, non-invertebrate system, we get better, more complete picture of the immune system total. So Tony is going to talk about this today, and then I'm very excited about his talk. And his talk is diversity and evolvability of self, non-self discrimination systems and origins of adaptive immunity. And
Thank
take a
you. Can everybody, I'm assuming everybody can hear me. I will share my screen. I will, hold on a second.
Sounds good.
Great. Can everybody see my mouse? I hope.
No, it's not sharing yet. Maybe give it another
Okay,
try.
hold on a second. Close this. Wait a second. There we go. Right there. Is that good?
Not yet.
All right. Wait a minute. How do I do that? Sorry, everybody. I want to be sure it's the pointer. Wait a second. Sorry about this. You know, anybody know how do I turn the pointer on? Hold on a second. Oh,
You're the co-host now. We should be able to see your pointer once you're in sharing mode.
okay, that's good. Okay, that's great. Is it sharing now?
Not yet.
It isn't. Okay, hold on a second. Here. Oh, here we go. I see. There we go. All right, here we go.
Yeah, then once you're sharing, we should be able to see your pointer.
How's that? Is that sharing?
Perfect.
Perfect. Okay, thanks everybody. I'm really excited to give you this talk today because we've made a lot of progress in the last year, and this is mostly due to this guy right here, Henry Rodriguez Valbuena. But I just wanted to give this acknowledgments first. But this is work from Spencer Nyholm and Marie Neidham, who were postdocs in the lab. Henry, who just was game changing in terms of how we're thinking about what I'm going to tell you about today, which is self, non-self recognition and the evolution of immunity, and Tanya McKittrick, who did some fantastic work. Okay, so what I started working on this weird system for, because I came, as Yuko said, I came out of mammals, and I was going to go do hematopoietic stem cell biology and immunity. And I was talking to Irv Weissman, and he said, you know, maybe you should think about this other system that I've been studying for many years. It's down at the Marine Station, and that was something I'd been familiar with. So I took it, and I'm going to try to explain to you and hopefully convey to you why this is such an exciting problem, I think. But what has motivated me the whole time is this question: what underlies the rapid evolution of immunity? Okay. And I'm going to give you two examples of that I think everybody's familiar with on this. One is this: how can cures have replaced live 49s, which replace something else on NK cells, but they all have the exact same function? How can this go on where you completely change receptor properties, right, extracellularly, but you still have the exact same function, which is missing self-recognition? And the second is, and more from Yuko and Martin Flannix, is this evolutionary mystery. So this is a jawed vertebrate. It appears in the fossil record about 425 million years ago. And it has almost every single thing we have in our adaptive immune system. It has B cells and T cells that are clonal. It has thymus. It has affinity maturation. It has memory. It has primitive lymphoid tissue, right? It has everything we recognize in us, right? It has the full package. You go one step down the evolution ladder to the jawless fish, which are also vertebrates. They appeared, well, about 130 million years before the split between the jawed and the jawless vertebrates, right? And they have nothing. There's no MHC, there's no BCR, there's no TCR, there's no RAG, there's no AID, there's nothing, right? There's no genes. And this is completely different than any other kind of evolution. So if you look at evolution of the kidney or the nervous system, you don't see anything that is this abrupt. The whole system came up. And most people I think most people on this webinar can appreciate the unbelievable interactions that go on. To make an antibody, you need T cell help, antigen presentation. None of these things are found, and there's no intermediate forms. And this is amazing, right? So what are the origins of adaptive immunity? Where did this system come from, right? And I'm going to tell you where it didn't come from. It didn't come from here, right? It wasn't just handed down. It evolved from something, right? There's really nothing, no extant animals that we can look in the genomes between jawless. There are not many jawless fish around that are still alive, and there's nothing really intermediate that survives, so we can't look in the genomes, right? But nevertheless, this is an amazing leap in complexity for an immune system, right? So obviously it evolved from something, and the question is what, and that's what I want to talk about today. Before I do that, I want to point out these 4 points and this. And what is the foundation of adaptive immunity? Okay. First is somatic diversification and clonal identity. And that is this. Each cell makes a receptor on its own by somatic recombination. And this is important. And the way I want to say this is specificity of each cell is not encoded in the genome. Specificity of the receptors on the cell surface are randomly generated. That's one. The second thing that sets us apart is education, right? And I'm going to refer to this throughout this talk as quality control. And this was a kind of a concept I talked to you about a long time ago with Thomas Bohm, and he wrote this beautiful review on it in Cell years ago. When quality control is what establishes a new interaction, a new specificity, and what maintains it, okay? What establishes specificity in the adaptive immune system is education or positive and negative selection, thymic education or in the bone marrow, right? And again, that's just telling you specificity is not encoded in the genome. Specificity is determined by an education process that goes on in the body. And I'm going to tell you that this is vastly different than what happens in all the other non-self recognition systems that preceded it. Okay. The third thing is clonal expansion. Okay. Anticipatory immunity would be worthless, right? If you think about how the strategy of the immune system is to create 100 billion receptors and find a needle in a haystack, right? One of them is going to work. But this is all dependent on clonal expansion, right? That there's a tremendous proliferative potential on each one of our naive lymphocytes when it gets stimulated, right? And this is the thing actually that I have found no intermediate to. This is really fascinating. Okay. Actually, that's not true. Hold on. I'll come back to this. And the last thing, what are the foundation of adaptive immunity? I want to point out is this. TCR discrimination, the way a TCR discriminates between self and non-self is really, really interesting and very different than the rest of the self non-self recognition systems. Okay. When a TCR binds to a peptide MHC in the periphery, it's comparing that signal to what it knows from the thymic education, right? It's comparing this. So if in thymic education, you're going to go through positive selection and negative selection, right? There's death by neglect and death by binding to site. And you hit the sweet spot of TCR affinity of the cells that come out. But for each one of those cells, right, their activation threshold is what's above the negative selection threshold in the thymus. Okay. So it's comparing two states. And the point here is that all non-self recognition by T cells is based on self-recognition, right? It's always in comparison. It's not just binding something, it's comparing in order to do that. And the question is, of these 4 properties, where did these come from? Okay. So I've been writing this review for interviews, and this talk couldn't have come at a better time because I it's really made me kind of narrow down a lot of my thoughts. So I've been looking through non-self recognition systems throughout multiple texts, from bacteria all the way up to us, right? So non-self recognition is a fundamental property. It's the whole there'd be no multicellularity without non-self recognition. And one of the most interesting things that I don't know if people are aware of is what goes on during neuronal wiring with 2 proteins called DSCAM and in the vertebrates clustered protocoadherins, which I'm going to talk about. Okay, but the functions of non-self recognition throughout the other texts are immunity, mate choice, social interactions, and detecting relatedness and neural development. Okay. Outside, and I know a lot of this is obvious, but just to be on the same page. Outside of the vertebrates, these systems are innate, right? So the genes that are involved in self-non-recognition are germline encoded. Okay. But the interesting thing is that we said there's no genes for the BCR, there's no genes in the TCR. These things are evolving just as rapidly. things just come up, there's new genes, and there's no kind of hint of where they came from, and there's this complex recognition system, and then that disappears, and another one appears, and this actually is repeated. There's really not it's been referred to by Martin Flanagan as a Big Bang in Yuko, right? And everybody and as the Big Bang is the origins of the vertebrate immune system, but actually that's quite normal. That systems that do self-non-self recognition just kind of appear out of nowhere. Okay. Finally, there's really three strategies in innate recognition there's three strategies that innate recognition systems use to discriminate. Okay. And I call them self-lock and key. Okay? And there's also homotypic, and I'll explain that in a second, non-self-lock and key. These things are basically the same, and I'll show you these in the next two slides. And then the one that's very interesting, that's been around forever from bacteria, is missing self-recognition. So most of the self-non-self recognition systems that are in multiple taxa are actually allorecognition systems. And these systems have evolved to do two things: either detect the presence or absence of a self pattern, okay, like an NK cell looking for MHC, or an inhibitory receptor searching for an MHC, right? Or what was I going to say? I'm sorry. Okay, I'm going to skip to allorecognition. Or I'm sorry, or changes in a polymorphic ligand. Okay, so either you're looking for presence, absence, or changes in a polymorphic ligand. Those are allorecognition systems throughout the rest of the taxa. And I want to explain this now, one of these, because this kind of explains everything. So this is an innate, what I would call lock and key system. This is self-incompatibility in the breast case. So this is broccoli. Okay, so self-incompatibility is an allorecognition response. Okay, and it serves to prevent inbreeding in hermaphroditic flower. And what happens is on hermaphroditic flowers, you have the pollen and the pistil, so the male and the female. Okay. The pollen is expressing a gene called SCR or SP11. The stigma is expressing a gene called SRK. Okay. If a self pollen lands on a self flower, they express these two proteins that fit together like a lock and a key. And if they come together, that initiates a signal transduction pathway that blocks the pollen from fertilizing that flower. Okay. So this is blocking inbreeding. If they do not match, then you can cross. Okay. So this is going to prevent inbreeding and the cost of inbreeding. That's the evolutionary pressure that's causing these to evolve. Okay. In most populations, there's about 30 lock and key specificities. So 30 alleles of this system, and there's about 100 worldwide. Okay. And what is very interesting is this. In this case, and in all the innate systems, the lock and the key must be linked in the genome. The alleles of any specificity must be co-inherited for it to work. Okay. And because of that, you want to prevent recombination between them. So the lock and key must be protected and encoded in a haplotype. So these are the three haplotypes of the brassica lock and key system. So here's the SRK, and this is the SCR or SP11, both of them. And you can see how different these haplotypes are, right? They switch genes around. The intergenic regions are completely different, so they can't recombine. So in other words, what happens when you make a new specificity, right? How does this happen? And this is amazing, right? So you can imagine you have a lock and a key. Okay. How do you get a new specificity? So say I need more recognition ability out in this population because there's too much inbreeding. So there's a selective pressure for that. How does that happen? And this is really crazy, right? So in one individual, a mutation happens in the lock. Okay. And two things can happen when that mutation happens. Either that lock can now become non-functional, right? And if it's non-functional, then it can inbreed, right? Because the lock and key no longer fit. Or it can be promiscuous and bind other things. So now you would still have blocking of inbreeding, but you might block some outcrosses too, okay? So to get a new specificity, a pre-existing specificity must be broken, okay? And because these are innate, it's through germline mutation, just normal germline mutations, okay? Happens, and you change the specificity, right? In order to get a new interaction, though, you have to have a complementary mutation in the receptor such that they fit together, okay? And more importantly than that, and more remarkably, and I still can barely wrap my head around this, the two have to occur on the same DNA strand to work, right? The same genes that are linked together have to have complementary mutations such that they bind together. It's almost impossible to imagine that that can happen, right? And I think this is because I just don't appreciate time and chance, okay? And once there's a new specificity, right, it's got to be protected. But the important point here is this. When you make a new specificity, how is it tested? How does it evolve? It evolves in the population. The selection is on the individual that carries that first mutation, right? And if that individual starts inbreeding, it's going to be this is going to be negatively selected in the population because this is going to have reduced fitness, right? And until that complementary mutation comes, and it has to occur on the same DNA strand. So the selection, when you think about thymic selection, now the selection in this case is out in the population, selections on the individual, not on the cell in the thymus, right? So this is completely different. Okay, so these are quality control mechanisms. Okay, so in innate systems, diversity is generated by germline mutation, selections on the individual, specificities encoded in the genome, right? The lock and key are in the genome, the specificities in the genome, right? And that you get about in all of these systems, I'm just giving you one, but in all these systems innate, you get about 100 in the population, in the world. Okay. So you can imagine how hard it is since you're waiting for two complementary mutations to occur, not only in two genes, but on the same DNA strand, right? You get about 100 lock and specificities through these populations. And there's all the kinds of things for the MHC evolution people out there. There's trans-species polymorphisms. There's everything like that. Because once you've got one, you want to keep it. So my question is, how did we go from that to somatic recombinations to recombination occurring in a cell, thymic education or central tolerance in B cells, which is essentially negative selection, getting rid of autoreactive, and going from 100 to 10 to the 11th to 10th and 15th, right, for ECR and the TCR, right? How did you jump from an innate system where evolution is going on and selection is going on the individual in the environment to internalizing these to inside a cell where you diversify cell autonomous, somatic recombination, you test that cell in a protected environment that has no cost to the individual. Like, I don't care. Most of my cells die by death of neglect. What do I care? Right? That's no cost to me. I'm not under evolutionary selection at that moment for that. So what are the intermediate steps? And it turns out there's 3 of these from those 4 characters I showed you. Okay. First of all, is somatic recombination unique? No. Somatic recombination has been around forever. And the best example of this is tetrahymena mating types. Okay, so tetrahymena is a ciliate unicellular. Usually unicellular. When you stress them or starve them, they mate. And this is a common theme in self, non-self recognition too, that I just want to go on and aside. Detecting stress. Every time you see self, non-self recognition, there's some detection of stress. Whether it's I'm starving and I need to mate, or I'm trying to detect cheaters in the population. It's absolutely amazing. Stress and self, non-self recognition like match together. But anyway, so tetrahymena are going to mate, okay, when they starve. So they're usually living as unicellular, and this occurs in a bunch of animals, and they come together to mate. And when they do, they need these mating types to meet, and this is to prevent inbreeding too. Okay, so there's 7 mating types in the tetrahymena population, and this is thermophilia. And this is work by Ed Arias, who was here long before he retired at UCSB. And he discovered this, and this is really cool. So they have 7 mating types. And the way the mating types are called, they're lock and key, but it's what I call non-self lock and key. So 1 will bind 2 through 6, right? But not 1. So when you have a lock and a key, it means you're not binding yourself. So it's not homotypic binding, it's enforced heterotypic binding, if that makes sense to everybody. So if you have 1, you can breed with 2 through 7. If you have 2, it could be 1 and 3 through 7 and so on. So each mating type binds every other mating type but itself. And this actually is seen in multiple systems too. Okay, so the question is, how do you determine mating type? And this is this crazy story. So and somatic recombination is central to the biology of ciliates. And there's these het proteins that people might be aware of out there. The same thing happens. But basically in tetrahymena, there's two nuclei. One's called the micronucleus, and that's like the real nucleus. And then when they start to grow, they have a macronucleus. And the macronucleus is it's fascinating. So they take all the genes, so they cut out all the stuff that's the junk DNA, and they just have the genes that are encoded. And I'm simplifying this a little, but it doesn't matter. And then they amplify those, right? And that runs the body of the cilia. But they keep this reserved micronucleus. So when they make this macronucleus, they're going to make one of the 7 mating types. So they pick one of them, right? And in the germline configuration, in this little germline, all of them are together, right? But in the macronucleus, they only have one. So what they do is somatically recombine there's different theories about how this goes on, whether it's intramolecular or intramolecular. So if it's going on within the DNA strand or between DNA strands, doesn't matter. There's a recombination event that gets rid of 6 of the 7, choosing one randomly, right? So each one, tetrahymen out there, if you starve them, chooses one randomly, and only the ones that choose one different mate. And it turns out 7's enough to prevent a lot of inbreeding. Okay. This is the other system that absolutely I had no real idea of, and it just blew me away. And this is just amazing. And I'm telling you about the second thing about non-self recognition, and this just shocks me. And I think everybody who studies NK cells and T cells will find this as exciting as I do. So this is a process called neuroavoidance. Okay, and the idea is that when we are assembling complex circuits in the brain, what you want to prevent are dendrites from the same neuron from synapsing with each other. And you want them to choose other neurons to synapse. And there's actually a non-self recognition system that is generated in each neuron. And like a T cell, each neuron is going to express proteins that make it unique. They're going to do a barcode. And how does this work? And there's 2 systems called DSCAM, but I'm going to talk about clustered protocoherence. So in flies, this is done by DSCAM. In vertebrates, this is done by clustered protocoherence. And this is how they get a non-self identity. So there are 3 types of clustered protocoherence called alpha, beta, and gamma, and they're clustered in the genome. Okay. And each individual stochastically expresses, so NK people should be familiar with this, stochastically expresses a subset of the clustered protocoherence. And each individual expresses 10 to 15 of these. So how does this work? So it turns out that each one of the loci will only do homotypic binding. Okay. So this locus and this locus and this locus, all of those will only bind to themselves. So basically what happens in the clustered protocoherence is each individual neuron on the surface of the cell expresses a subset or a repertoire of clustered protocoherent genes, right? And somehow when 2 neurites from the same nerve come together, those form into this huge zipper. And if one of the 10 to 15 don't match, they don't. And this is called the isoform chain mismatch model. So discrimination is made by some physical interaction between these. And what happens with these is that there are 6 domains on each cluster protocoherent. 1 through 4 bind in an anti-parallel fashion only to themselves, and 5 and 6 are promiscuous, right? So they express these things at the cell surface, and not all of the molecular details are known. The cluster protocoherents come together in cis, hold out their regions in trans, and only when the whole repertoire matches do these things bind together. And this zipper actually, and this is counterintuitive, causes the dendrites to break apart. So dendrites are discriminating self from non-self within an individual using a repertoire of clustered protocoherent expression. Okay. And what fascinates me is discrimination is made by some physical, well, this is what it's thought, some physical interaction between the repertoire. So basically, what's being recognized is a pattern between two individual dendrites. And if they match, that pushes them apart. If they don't match, that allows them to make a synapse, right? So in this system is innate, okay? The cluster procatenaries are encoded in the genome. Specificity is homotypic, so it's encoded in the genome, and they're comparing patterns. What's shocking to me is there's no education process. There's nothing. They just match or don't match. I still don't truly understand this physically, but there's this beautiful paper in Nature where you can actually see these zippers form. So the idea is if 1 of 10 of the isoforms they express don't mismatch, they cannot form this huge zipper that forms. And this is shown right here. So this is what would happen when two cells come together and they totally match, they zip. If there's two cells that aren't, they will not mismatch and this zipper will not form. This is a picture of what this zipper looks like, and this is pseudocolored here, right? So these are dendrites. So this is self-non-self recognition system used during development. Each cell has a unique identity, right? And then homotypic recognition blocks dendrites. So this is absolutely required for our brains to wire. Okay. All right. The third thing is somatic education of NK cells. So NK cells are innate cells, right? But they have an internal education mechanism. And we all know that NK cells function, right? And do almost everything that I just said to you. They have a group of receptors. They stochastically express them. Each cell then has a random identity, right? They use these receptors to discriminate healthy cell from an unhealthy cell, and they using two types of receptors. One is they're looking for the presence of a health ligand, and if that health ligand disappears, those cells are killed, or they're looking for overexpression of an activation ligand, right? And if that's overexpressed, then they kill those cells. So it's a balance of inputs that determine whether NK cell works. And this is exactly like T cells, right? In order to do that, you are using a self as a reference, and that self is determined just like the T cell during development, during education, right? So you know it, that NK cell knows what normal self is based on the receptors it has stochastically expressed. So specificity is not encoded in the genome. Specificity is determined by an education process, okay? So like the T cell, the NK cell detects changes ligand density using self as a reference, and it is also, right, randomly expressing activating and inhibitory receptors. So it's making unique cellular identities. So if you have stochastic expression, and one thing I would love to talk about is why is that? And is this a thing where you have two, why don't you express all your receptors, right? And you can still discriminate, or do you express a subset of receptors because that makes you more sensitive to change? And I'll show that again. But no matter what, you express receptors at random, so you have to set your activation threshold, right, based on the receptors you just expressed. There's no way the cell could know what that is until it's expressed receptors. So education is required, and that goes on during development, right? So this is unlike the self-incompatibility I showed you, where the selection's on the individual, selection's on the cell. And I know everybody knows this, but I just want to put this in context, right? And what do we know about NK education is that during development, an inhibitory receptor must bind its ligand for a cell to become functional. It's called licensing, but I don't think of it like that. So I want to tell you how I think of it. So I think of education in terms of the rheostat model, right? And I think this. So let's say you've expressed 3 inhibitory receptors, right? And you have the inhibitory ligands for them in your body, right? So you have 3 inhibitory inputs, right? The goal of the NK cell is to determine when one of those genes, one of those proteins has disappeared, one of its inhibitory ligands has disappeared. So it needs to set the threshold for activation right below the highest, right, endogenous inhibitory inputs it would have. So if you have 3, you'd set your activation threshold here. And if you only expressed 1 inhibitory receptor and had 1 inhibitory interaction going on, you would set your activation threshold here. So that's how I look at the rheostat model, or this is how I think about the rheostat model of NK cell education, right? Because you're balancing the two so that you can so you can detect changes out in the periphery, right? And I'm also going to say that I think licensing and education are 2 different things. I think education is just like what a T cell does. T cell goes in, say it binds a little too tightly, it might downregulate LCK, it might upregulate SHP, right? It's going to tune its binding, right, to try to get into that sweet spot. And I think this is the same thing that goes on in NK cell. And I think licensing is something different. I think licensing is the same thing that happens for peripheral tolerance or for B cells, right? Is that you become anergic, right? If you don't get that inhibitory response, you become anergic. Yeah. All right. So given that, I think immunity evolved from natural killer cells. I think adaptive immunity is there. I think everything about an NK cell is exactly like a T cell. A T cell just has taken it one step further, and the acquisition was not any of the education or anything like it was the acquisition of RAG and allowing it to diversify. So how do I think about this is the Red Queen, right? And this is again, missing self-recognition as a form of immunity is found in bacteria. Okay. It's been around forever. Okay. So any lack of a self pattern can be a sign of stress and that can be interpreted by the cell. So bacteria is this thing called toxin antitoxins. And when this is blocked, this is a phage, often come from a phage infection, and they respond to that. Right. So missing self-recognition is ancient. Okay. So if you imagine there's missing self-recognition and that some of the first immunity in the protovertebrates or whatever are looking at self-ligands. And if the self-ligand's downregulated, that's a sign of stress. It's like it's a damp, right? So an inhibitory ligand's downregulated, that's a sign of infection. Okay. So what do we know happens? We know that viruses evolve decoy molecules to fake those inhibitory receptors out. Okay. And now you're in an arms race, right? And now you're forcing evolutionarily the inhibitory receptor to become polymorphic and be able to discriminate from the decoys. And this just goes on and on. All right. And you have this paired receptor system, and that's evolved. And I'm going to tell you that has come previously too. Okay. So you have this in an innate sense going on, but you have education. So you have this evolvability of the receptors that we know occurred, right? We know these receptor families have evolved from zebrafish are different than the mice and rats and are different than the primates, right? So missing self. Okay. So you have this arms race going on. Now RAG comes in, which everybody thinks this is the origins of immunity, RAG jumped into a receptor. My opinion is that it had to be an inhibitory missing self-receptor because RAG jumps in, right? And then you have all this diversity, but NK cells already know how to deal with diversity. They already have an education process. So this is the precursor, I think. And then peptide presentation evolves after that. But already, the NK cells are recognizing class 1, and I think peptide presentation then evolved next, right? Because it's another sign of stress, right? And this already goes on because this is all about ER stress when cells are infected too. Okay. So I think NK cells are just innate T cells. They do everything T cells do, but in an innate way, right? So they have somatic diversification. That's in T cells by somatic recombination. That's stochastic expression, but cells are clonal. Okay. Education's the same exact thing, right? We couldn't have cures replace LID49s if there was an education. Education doesn't care what the property of the receptor is on the cell surface, only that it's integrating the right signals inside. NK cells also do clonal expansion, right? And that's kind of a newer thing, but that's the other characteristic. And NK cells are doing the same thing at TCRs. They're comparing two states, a healthy state from an unhealthy state, right? It's just that the TCR is one receptor doing what a bunch of activating and inhibitory receptors do in an NK cell. It's all been condensed into one recognition event, right? Which is a recognition of self. So TCLs recognize self because NK cells recognize self, and peptides just came last. That's what I think is going on. And I'm just going to finish up now talking about where did those properties come from? Where did education come from? Where did stochastic expression come from? And this is where the work from our lab comes in. Okay, so I work on allo recognition in what's called a tunicates. Tunicates are a sister group to the vertebrates, so they're chordates, and they're chordates because they're born a chordate tadpole, which swims around. These are marine organisms, swims around for a little while. And this is a chordate. It has a notochord, it has a hollow nerve tube, everything about chordates, which we are also chordates, swims around, settles, metamorphs into an invertebrate body. Okay. Then that invertebrate, this is a special kind of tunicate called a colonial tunicate. Not, this is somewhat important, but not too important. But basically, what happens is that once this guy lands, it starts this asexual reproductive process that eventually give you rise a colony of genetically identical individuals. Okay, these are all linked by a common blood supply. Okay, and the orange you see here are pigmented cells in the blood. And if you flip the colony over and inject the vasculature, so there's a huge extracorporeal vasculature, and you inject it and it runs throughout the colony. And you can see this here. So this is these bodies, which are called zooids, and this is the blood vessels. And then at the edge of the colony, the blood vessels terminate in these little protrusions that are blind-ended, sort of sausage-shaped structures called ampullae. Okay. And this is a live image of these vessels. So these things out here, so here are the bodies, and these little things out here are the ampullae, which are just termination of the vasculature. Okay. So these guys go as marinos throughout the world, and this is what they look like, and we can collect them and bring them in. We also have inbred strains, okay, of these guys, and this is how they grow. So they're great for imaging. So they go from a larval tadpole to a sessile adult, right? And we grow them in the lab. Okay, so you can imagine this tadpole lands and it starts to asexually expand outward. And in where these animals are, there's incredible competition for space. Okay, and when they bump into each other, the first thing to touch are ampullae. And when the ampullae touch, this initiates a natural transplantation reaction between them. Initially, there's an inflammatory reaction. So this is two colonies touching. They're blue, and you can see the blue. These are the pigment cells in the blood. They come together, okay, and they touch. And one of two things will happen. Either the two blood vessels will fuse, okay, and now two individuals will share a common blood supply and they form a parabiosis, or they reject each other. And this is an inflammatory reaction where these cells called Morula cells pour out between the two interacting ampullae. They explode. In them are the precursors for this thing called prophenol oxidase pathway. And these make melanin scars, and that's a rejection response. So they touch, and within 48 hours, they fuse or reject. But why is this of any interest? Well, I'm sorry. One other thing I want to tell you is in the ampullae, these allorecognition reactions are spatially segregated. Okay. So everything that is occurring, the recognition is occurring on the tip of the ampullae. So an animal can fuse to a compatible colony on this side while simultaneously rejecting on the other. So everything is spatially segregated, and that will become important in a second. Why is this interesting though? Is the genetics. Fusibility is controlled by a single locus that's called fusion histocompatibility. It's one of the most polymorphic loci ever described, certainly the most polymorphic innate locus. Okay, there's about 300 alleles in any population, right? It's about 1,000 alleles worldwide, meaning there's a self-non-self discrimination system that can discriminate between 1,000 alleles of the polymorphic ligand. And the rules of allorecognition are really interesting. If two ampullae come together and they share one or both alleles that are compatible and fuse, if they share neither, they reject. Okay, so this is classic missing self-recognition. When two ampullae come together, they're looking for self on the other ampullae. If they see one or both self, that signals compatibility and fusion. If they see neither, they reject. So this whole system is based on detecting self. Okay. The other thing is colonies don't reject things they bump into. They only reject under other members of the species Botryllus schlosseri. They don't reject other species. They don't reject rocks. So there's a species-specific activation signal, okay? And as I said, it's spatially segregated. So why is this relevant to archaeobiology? Because discrimination is carried out by germline-encoded receptors, just like the NK cell, right? The outcome is also, as I'm going to show you, a balance of activating and inhibitory signaling, okay? Two families control whether they fuse or reject. They're called Fester and the Fester co-receptors, and they use classic ITAM and ITAM signaling. Okay, and this is a thing I don't know and has kind of thrown me as I look at all these other systems. We think this requires an education process to establish specificities, because I don't think you could ever explain 1,000 specificities as being genomically encoded. but I don't know if that's true. Okay. And we also have evidence of education I'll talk about. So the question is, what endows an innate recognition system, with a discriminatory ability that we're more used to in terms of the adaptive immune system? How does a system evolve that can discriminate between 1,000 alleles? Okay, so I'm going to tell you how I think that works. And I'm going to tell you why we think that. Okay, so go back to NK cells. What inhibitory receptors in the primates, the KIRs, recognize 4 epitopes on MHC class 1, right? BW4, A311, C1, and C2, right? And when a cell comes up, it is integrating activating signals and the inhibitory signals to make a decision, okay? What we think goes on in Botryllus is exactly the same thing, but it's all focused on one ligand. So the ligand we call the FUHC-seq. It is highly polymorphic, okay? It has a bunch of different epitopes, and we think we express a repertoire of receptors that bind these epitopes, okay? And I think what happens is there's an education process that goes on cis. So this is the ligand, and these colored shapes represent the epitopes. So this is like the C1 or C2, however you want to think about it, right? And we have ligand receptors, which are heterodimers between these two that bind each of these epitopes, okay? If a self comes in trans, they all switch from a cis to a trans, right? And if they don't, only some of them move, okay? And all of them have to move in order to be compatible. That's what we think is going on. We think in the ligand, there's 20 epitopes, okay? And that these things bind. In addition, these have ITIM and ITAM domains. So the other thing we think is that how discrimination is made is basically the orientation and the 3D structure of the ITIM and ITAM domains that are on the receptors that are basically, when they bind like this, are basically representative of the distribution of the epitopes on the FUHC-seq. So this is all but testable speculation. And we think that somehow, whatever the amount of receptors that are expressed, right, go through an education process, and I'll explain that in a second. So why do we think that? Okay, so I know I'm running over a little bit. I'm going to jump through a bunch of this, but basically, as a postdoc, I positionally cloned the locus, right? And the locus is huge. It's about 800 kb and has both the ligand and the receptors. So the ligand we think is this protein called FUHC-seq. Okay, FUHC-seq is a EGF and has an EGF family. It's interesting. It has no transmembrane domain, although it's encoded right next to your protein transmembrane domain. And this is right here is called DIVA analysis of the alleles. So if we look at a bunch of the alleles and we look at where the polymorphisms are, all of these are very highly polymorphic. And the difference between any two of these FUHC alleles is about 20 amino acids. A lot of them have advantage positive selection, but they're scattered throughout the protein. So there's no hypervariable region. And this always bothered me when we first saw this. Like, how are you discriminating between this if there's nothing really consistent between the alleles that would give you structural information? Okay. We also found 2 receptors, and I'm going to go through this really fast, and I called them Fester and Uncle Fester. Okay, so Fester and Uncle Fester are type 1 transmembrane proteins that have an immunoglobulin domain and a sushi domain. Okay, Fester is polymorphic, and Uncle Fester was not. Both of them, these are monoclonal antibodies. They were expressed on the surface of the epithelium as well as a subset of the blood cells. Okay. And again, Uncle Fester's monomorphic, has the same structure, but the 2 genes are barely alike. That back end of the gene is almost 100% identical, but the front end of the gene has almost no amino acid homology at all. But they still form an immunoglobulin and a sushi domain. So there's conservation of the back end, but not conservation of the front end. Okay, so what we found is we made monoclonal antibodies to both Fester and Uncle Fester. Okay, and I want to tell you about Fester first. So we had so Fester's polymorphic, and there's about 60 alleles, nothing compared to Seq. FUHC-seq has about 1,000 alleles worldwide. Fester has about 80 alleles worldwide, right? So it's obviously not a lock and key interaction. Okay, so we made a monoclonal antibody Fester, and we did monoclonal antibody interference experiments during a fusion or rejection response. So if two animals were fusing and we put that monoclonal antibody to fester, it had no effect. But if they were rejecting and they both carried the allele we made the antibody to, we could turn a rejection into a fusion. Okay. So the interpretation of this is the antibody was stimulating the receptor that I would the analog of the inhibitory receptor. So the receptor that saw the self-ligand, we were overstimulating, and we could override a rejection and turn it into a fusion. We also made a monoclonal antibody to Uncle Fester and got the opposite result. In this case, so this is a great experiment by Tanya McKittrick. She took the monoclonal antibody, put it on magnetic beads. These guys are growing in the water. And she put the antibody on just regular magnetic beads, put them in, and she could touch the ampullae. And if you touch the ampullae with the Uncle Fester antibody, you could induce the rejection. And if colonies were fusing and we added the antibody, we could override a fusion and cause a rejection. Okay. So what we have is a balance of inputs. Okay. So we have an inhibitory receptor and an activating receptor. Okay. We can tune each in vivo and convert each reaction. So this is just like NK cells. You're measuring two inputs, right, and making a call. So I'm going to jump ahead here. Okay. So we knew there were two independent pathways run by two different receptor families, but this left us with three questions. How are the activating and inhibitory signals transmitted? What was the activation ligand? And how did the 80 allotypes discriminate between 100? Okay, so the Fester and Uncle Fester had no single transduction domains on them. So we didn't know how that happened. But if you FACS isolate the cells, they express everything that's involved in ITIM and ITIM signaling. So we just assumed there was like some sort of DAP10, DAP12, whatever out there that just paired with Fester and Uncle Fester. So maybe there was an inhibitory one too. So that'd be slightly different, but whatever, they were activating an inhibitory. When we looked in these cells, they have every single gene that's evolved in T and NK cells, ITM and ITM signaling, ZAP, Src family kinases, SHIP1, SHIP2, SHIP, etc. Okay, and all these are expressed in the ampullae. Okay. Then what we found is that we've been completely duped by this. So we thought we had two receptor families, and it turns out that wasn't true. And Henry found, and this was because of AlphaFold and because of the lockdown, he started looking at other genes that we found in the genome that kind of look like Fester. And we went back and modeled them. They all had the exact same. So these genes, the Fester genes, so we found a bunch of Fester genes that are expressed in the locus, okay? And they're completely divergent from each other. The pairwise amino acid homology between them is about 30%, but they all make the same exact structure. So this is an overlay of the immunoglobulins, okay? To put that in context, this is the pairwise amino acid of the different human KIR loci. Okay, so these are very, very divergent even compared to KIRs, okay? The other thing, and then the next thing we found is encoded next to each of the Fester was a new gene that we'd also missed because it was such low homology. We call this the Fester co-receptor. It's a type 1 transmembrane protein. It has 3 immunoglobulin domains and a sushi domain, 1 transmembrane, and inside there's F co-receptors have either hemITAMs, ITAMs, or ITAMs. Okay, so we linked this now to the signal transduction pathway. Okay, and the last thing I want to tell you before I finish this up is this, that we've also found that these different Fester co-receptors can swap their ITAM for ITIM domains by alternative splicing, and we see this in different individuals. So different individuals will have the same ectodomain, but by alternative splicing, and this is shown right here, there's 2 of these genes called FCO4 and FCO3, and they alternatively splice their ITIM and ITAM domains. Okay, they're activating inhibitory. So a receptor in an individual can change whether it's activating or inhibitory. Okay, there's a bunch of these encoded. The other thing that we found is that in the FUHC locus itself, where this is the ligand, there's tremendous diversity in different haplotypes of the Fester and the Fester co-receptors. There's also another locus where there's another set of receptors, Fester and Fester co-receptors. They are encoded in pairs, birth and death evolution goes on in pairs. So the difference between two haploids, so if you think of Kier's and Kier loci are different, in this case, pairs of Fester and Fester co-receptors are different. Okay. And this is what brings us to the final thing. So what we think is there's pairs of activating inhibitory receptors binding these epitopes, right? And this is how discrimination is made. So each cell expresses a Fester and Fester co-receptor repertoire that is somehow educated to the epitopes, right? That the way discrimination made is the same way an NK cell makes discrimination. It's by balancing inputs, which we've shown, right? And that this is the intermediate step prior to NK cells. So the way to think about this, and I talked to this guy, Robert Bush, a few weeks ago, and he gave me this, and I've been thinking about this, and this is great. So if innate systems are lock and key, right? Botryllus and I think NK cells are like a combination bike lock, right? So you need the right combination in order to open the lock. Okay. That's what we need on this system. And this is remarkable, right? Because to make a new specificity in these innate systems, you have to break a pre-existing one. But in Botryllus, to make a new specificity, you've only got to change one of the dials to make a new combination. So this changes how fast it can evolve, right? And I think this is telling us that you have to somehow know the cell that each individual needs to know. So that's why I think there's an internal education process that goes on. So this is what I've been thinking. This is my last slide, and I'm sorry I've run over. Okay. But the question is that I've been thinking about. So now that I've told you about these clustered protocoherence, make a repertoire, there's homotypic binding. There doesn't seem to be any education. The question I've been thinking about is, can specificity in Botryllus be encoded in the genome, right? Can you have the right Fester co-receptors for that? And the thing is, we don't know right now because we haven't proved all the stuff I showed you functionally, but we have receptors in two different chromosomes, so they can't be inherited together. So it can't be lock and key. There's nothing that says it's lock and key. So I think that indicates there must be education, but I don't have any evidence of that. Okay. And the other thing I always think about is if Botryllus, right, can discriminate between 1,000 ligands, why aren't NK cells better at discrimination, right? Why are there only 4 epitopes they use? And the question for me, then what I've been thinking about a lot is, what's the role of these? Is NK specificity sort of restricted in evolvability just because of its role in pregnancy and immunity, right? That you just can't really redo that very fast, but Botryllus can, and Botryllus can because it has no role in pregnancy and immunity. The role Botryllus has is to ampulate touch and effuse or reject, right? So there's no pleiotropic things going on with this recognition. So it might make it just that much more evolvable. Okay, thank you very much. I'm done, and I'm happy to take any questions if anybody's still there.
Thank you very much, Tony.
Yeah.
I guess I have a question, but let's see. Martin has a question.
Okay.
What limits the size of Botryllus colonies? Is it ability of the vascular system to reach?
No, they can be gigantic, right? I don't know what does it. Probably we've seen colonies that are, I don't know, 200 centimeters across, consisting of tens of thousands of individuals. Yeah, so it's probably the blood and how coordinated they can stay. In theory, nothing limits it, right? And I know that because in the lab, is my screen still up there? In the lab, we can do this thing where we cut them in pieces and continue to grow them. So let me show you a second. So they form, let me show you, let me go back here. So these are the bodies, and they form into these little flower-shaped structures we call systems. And you can cut these into pieces and separate them, and they'll continue to grow. So we can have 100 pieces of the same genotype, right? So I don't think anything really restricts it, although they do have a lifespan.
So I get to ask questions. So
Yeah.
you mentioned about the education. So is the education putting item or item? Is that part of the educations?
Yeah, I think so. We think so, right? So what we see is that there's genotype specific expression of the festers of the F co-receptors, and there's genotype specific expression of the ITIM and ITAM alternative splicing. So in other words, one individual will have this receptor called F co-receptor, and it'll be all of it will have an ITIM, and another will have half ITIM and some ITIM, right? So and this is maintained. And the other thing I wanted to show you, hold on a second. I didn't
But that
show you this. Yeah. So
item,
one thing, what's that? Go ahead.
item switches, right? Alternatives.
Yes, it switches by alternative splicing. So
But
each
what's
individual,
causing that?
what's that?
Is there any cause? Like, is there any triggers to switch?
I think it's education. I think that during development, that's why we think it goes on in cysts too. I think during development, they're throwing up receptors and figuring out what they've inherited, what epitopes there are, and that's where the education goes on. So they know, right? Just how does an NK cell know where its threshold should be? Right? How does it know? How does a T cell know what a negative threshold is? Nobody knows that, right? And I think the same process is going on in Botryllus in that they're setting a threshold, right? And that threshold is they know what it's supposed to be. So they set the inhibitory and activating. And in this case, the other thing that's really cool about this is it's not inhibitory and act. Don't think of it as inhibitory and activating. It's integrating two signals. It's using two independent signals, right? And this is a thing called coincidence detection, right? And it's using two independent signals. And by using two independent signals, right, to make a decision instead of one signal that's on or off, you have all the tunability of the system, right? So I think that's what they're doing when they set up when they're initially doing it, because they're discriminating. It's hard to imagine lock and key because all the lock and key systems only have about 100. And then all of a sudden in Botryllus and other L recognition systems we haven't talked about, there's 1,000 specificities. So what is that leap? And I think it's education. I think or somatic. It's somatic. It's how an activation threshold is set. That's what I think has happened in Botryllus. And I think this is and honestly, I think this is the Big Bang. I think this is the most important thing. The thing that everything is founded on for both NK cells and T cells is internalizing specificity, right? I'm sorry, internalizing the quality control that I would say that establishes specificity. So specificity is established in these lock and key systems out in the environment. So if you got a crappy one, you die. When you internalize it, if your NK cell doesn't express an inhibitory receptor, well, it goes anergic. Of course, NK cells have another function, anergic, but the point is that, right, it's the same exact system that's setting up specificity has become internalized. And I think that is the huge evolutionary leap that everything else is founded on. And it is already in NK cells, so it must have come before NK cells. That's sort of my thesis today.
I have one last question. So there's only one pair of Vesta family and the co-receptor per
No,
cell.
there's many of them. Yeah.
Nice.
Yeah, right. Each individual has about 10 pairs.
And so
Right?
does
And they're random. Each haplotype has different. So I think there's 50 fester loci. There's 75 co-receptor loci. They form specific pairs and then different pairs. So the haplotype diversity is in the gene pairs, not in and that's shown right here. So these are gene pairs right here. So these are different gene pairs. So we actually have one. So we just started this. And one of the greatest thing at the SIP meeting was all the long read sequencing. So we need a lot more of those. But one individual got sequenced and it was so good. We got both chromosomes. So we have both haplotypes of all the chromosomes. So we know this haplotype diversity is really, really high. But what fires me up is really exciting, especially compared to cures, and I'd love to hear this from people, is that the gene content variation is not of a fester or a co-receptor, it's of both. Right. So when we compare haplotypes, although we've only compared to full haplotypes, we compare haplotypes, right? The copy number variations is of the gene pairs.
So it's not variegated expressions.
Right.
It's
There is variegate. Well, there's difference in, yeah, we have, yeah, here's the problem. This animal was never, no RNA sequencing exists for that. So I can't tell you if there's variegated expression yet. Yeah. Yeah.
so the multiple things controlling positive, negative signals, and then the balance between them.
Yes, that's exactly
Yeah.
right. That's exactly what we think. Yeah, exactly. I think that's
Any other questions? I don't see any in the chat. Anybody who has questions, you can unmute and ask him.
Oh, there's one that just came in.
Oh, just give me. Okay.
Okay, so the question is the current idea that the KIR signals are equal in the inhibitory and activating sense? No, I think, no, they're balanced, right? Is the way that I think about it, right? And then you have to set this to view to autoimmunity, you have to set the system up such that the inhibitory is just over the endogenous activating, right? So you need the inputs to set your threshold, okay? And that input, that education process when that's going on has to be from endogenous activating ligands, which nobody knows what they are, right? As of now, as of the last NK meeting that I went to this year. So I don't know. So detunica larvae, we're just doing that right now. Yeah, exactly. So this is a great question. This is from Steve. Yeah, so this is really cool, Steve. One thing that's really neat is when the tadpole settles and metamorphs, right here, it's competent to do non-self recognition. So if an education process has occurred, it had to occur in the embryo. So this is really a place for us to look at that. Do I think glycans play a role in these responses? No, not that we have any evidence for, because it all can be explained by polymorphism on the genes. Although I have to tell you, the pattern recognition receptors and these are really interesting. I'm sure they're doing like glycans. Anything else? Have I looked at allele frequencies? Yes. Oh, yes, we've looked at allele frequencies. So Marie Neidham is now a professor who was a postdoc, and she's done all this. We published a lot of the evolutionary work. Did they follow a power law? I don't know what that means. I'm so sorry. I'm a cellular guy. What is that? Can you? Does anybody know what that means? Martin, what does that mean?
Oh, I meant you have, sorry, I froze for a second. There's
Okay.
individual alleles, and you said there were like so many alleles that have been described in this colony and more worldwide, but how frequent are they? Are the same alleles showing up, or are they?
Yeah, so this is really cool. Yes, the same alleles show up. And part of this positive selection is that there's more diversity within a population than between them, right? So we do see all of this. And so Marie published this really nice paper on the evolution of these genes, looking at different populations throughout the world. So most of them are at low frequency, right? There are shared alleles between populations, right? But there's more diversity in the population than between.
Who wrote that paper? Sorry.
Marie Neidham, Nydam.
okay.
So she's done a bunch of this stuff and she's still
Oh,
doing
yeah. You mentioned her.
Yeah.
Well, thanks. I think we're at and overtime.
Yeah, I'm sorry I ran over.
Thank you.
The new talk. So
at least we gave you more than 15 minutes this time.
yeah, exactly. Really.
And well, thank you so much, and thank you.
Thank you.
Moderating.
Thank you for listening.